Hi, welcome to Tercen. My name is Martin. Today we're going to get familiar with running a project. We'll set up a workflow and visualize our data with projections. Then we'll make a calculation on the data and visualize the results of that. Let's get started. We're going to recreate a study on rock crabs done by Australian biologists in 1974. The local crab population had colour variations, but were always thought to be the same species. To prove a hypothesis that they were not, the researchers began taking morphological measurements. They compiled data on the characteristics of the crab's carapaces. What we find most interesting about this study is that they use a simultaneous mathematical analysis of multiple characteristics to separate the species. Our tutorial is going to show how easy it is to do these type of multivariate comparisons using Tercen. We will also take a look at how to use a statistical technique called PCA to simplify data and make it easier to analyze. First, some housekeeping to get set up. Install the PCA Operator to your library. Then create a new project on your personal page. Next, you'll need to download the example data file. It's on our GitHub. You can find the link to it on our crab tutorial document. Then we need to add it to our project. Let's take a moment to review our dataset so we can understand the information we want to analyze. There are 1000 records made up from 200 crabs that had 5 carapace measurements each. Observation is the number that identifies a crab, 1 to 200. Colour is orange or blue. There are 100 crabs of each. Sex is male or female, and there are 50 of each colour. The combination of colour and sex makes a natural grouping that we'll look for later. 50 blue males, 50 blue females, and so on. Variable is the label given to the part of the crab carapace that was measured. FL is frontal lobe, RW rear width, CL carapace length, CW carapace width, BD body depth. Measurement is the recorded size in millimeters. Okay, let's press upload now. Now that our project has a dataset, we can add a new workflow. The first step in any workflow is bringing in the data. Right-click the white background, press Add, and select Table. You see our dataset is available. Now we can attach steps that perform analysis. Let's attach a step to make a heat map visualization. Right-click on the table and add a data step. This screen is called the Cross-Tab Projection screen. Visualizations and calculations are done here. Click the plus icon beside our dataset. Now you see the headers of the data file: observation, colour, sex, and so on. On the projection crosstab screen, these are called factors. A factor can provide data to the visualization or affect how other data is visualized. It depends on where you drag and drop them. Pick up a factor with your mouse and drag it. You see zones that go green to tell you where it can be dropped. Different zones do different things when you drop a factor onto them. This element of the screen is called the crosstab grid. It creates the visualizations. On the inside, it has zones for X and Y axis, and outside them are the zones for row and column that create a multi-factor axis. You can drop factors into any of the zones. While there can be only one X and one Y axis, multiple rows and columns are allowed. Tercen will display their values in the crosstab cell. It applies a sort order working from the outside in, and within the crosstab cell itself, Tercen sorts values from low to high. One of the advantages of Tercen is that, unlike a spreadsheet, multiple values can be held in a single cell. Let's clear this simple projection and take a look at a heat map. We'll arrange the individual crabs along the top by moving the observation factor to the column zone. We'll put measurement on the y-axis and we will drop variable on the row zone. This will group the measurements by the carapace characteristic. Now we have an overview of the data that follows standard biology conventions. We can change how our visualization looks by dragging the lines of the grid closer together. Now we can see the 5 variables measured on our 200 crabs, all projected on a graph. This is the configuration panel. It has setting zones that control how data is visualized. If I drag the measurement factor to colors, Tercen will apply a gradient to the values in the crosstab cell. Higher values are assigned to hot colors and lower ones to cold colors. Then, by changing the type drop-down, our point graph becomes a heat map graph. I'll point out now that you should save your work regularly. Tercen will let you know a change has been made by displaying this disk icon. Click on it to save. It's also good practice to label your workflow steps clearly so that people can understand your research. I'll call this one Heatmap. Now let's do a visualization to see if we can reveal our crab species. We'll try a simple comparison called a pairwise plot. This way we can compare the crab's measurements and see if any pattern emerges between blue and orange. First, attach a new data step to the table. Then, in the projection, put measurement to the y-axis, variable to column, and variable also to row. Put observation on labels. Drop color onto color. That'll separate our suspected species. We've just made a multi-pairwise comparison graph. All the variables of the blue crabs are plotted against all the variables of the orange crabs. But there is a problem. There's no clear separation between the characteristics of the crabs that would show that they were different species. The dots are very closely bunched. Even if we split the graph by sex, easy to do in Tercen by moving the factor to columns, we still don't see any conclusive proof. The males do show a slight pattern of divergence in the carapace width, CW, but this projection of the data does not reveal the species. We'll have to make some computations on our data in order to gain better information from it.
In Tercen, computations are made by operators. We will apply an operator called PCA, which we added to our library earlier. PCA means Principal Component Analysis. PCA transforms the original variables into new variables called components. Projecting these new components allows for patterns to emerge that can't be seen with the raw data. Let's duplicate our original visualization and add the PCA operator. Run it and check its results here in the Computed Tables link. See, it has identified 5 principal components and calculate the value for our 200 crabs. Now we'll add a downstream data step to use the results for PCA calculation. In Tercen, the results of calculations performed by an operator and the original data are both passed down to the next step for visualization. See that our original factors are still available, and the results of the PCA calculation have been added as new factors. I'm going to pick up 2 principal components and compare them pairwise. We'll plot PC2 and PC3 into our axis, and put both sex and colour into the colour zone. Tercen will combine them into a group for the gradient. Now we can see the possibility that these are 2 separate species. If these crabs were the same species, the dots would be closer together, like they are on our multi-pairwise plot. Now that we're encouraged by these findings, we should compare all of the principal components against each other. It's possible in Tercen, but we'll have to wrangle our data into shape first. We need to group the components together so that they can be used as a block in the next projection. Working from the PCA operator, we add another step, this time a gather. From this name, Tercen will make 2 data groups, PCA variable for the names and PCA value for the calculations. Then we select the values we want to use. Let's pick all of the principal components. After we save the gather, we need to run it in the workflow. Now we can attach a new data step to visualize it as a multi-pairwise. To make this projection, place the PCA.value onto both the X and Y axis, then plot PCA variable onto both column and row. Crab color and sex are color coded as before. Drag observation to labels. This will separate the data points into individual crabs. When I adjust the grid a little, we get to see each principal component plotted against all of the others. Now it's visually obvious where the species separation occurs. Well done, you've created a workflow that visualized the crab's data and showed that a multivariate analysis of their carapace characteristics can separate them into two distinct species. Lastly, I'll add an export step, so my calculations can be downloaded for review or used in other projects. See, now both my original and my computed data are available for download. If you want to learn more detail about how PCA works, or how to wrangle data into a different format, check out our other videos. Thanks for watching.